Although keratinization occurs in fishes, among terrestrial vertebrates it becomes a major feature of the integument. Extensive keratinization produces a prominent outer carnified layer, the stratum corneum, that resists mechanical imbrasion. Lipids are often added during the process of keratinization or spread across the surface from specialized glands. The cornified layer along with its lipids increase the resistance of the tetrapod skin to desiccation. Multicellular glands are more common in the skin of tetrapods than in the skin of fishes. In fishes, the mucous cuticle and secretions of the unicellular glands at or near the surface of the skin coat it. Among the tetrapods, multicellular glands is usually reside in the dermis and reach the surface through common ducts that pierce the cornified layer. Amphibians are of special interest because during their lives they usually metamorphose from an aquatic form to a terrestrial form. In most modern amphibians, the skin is also specialized as a respiratory surface across which gas exchange occurs, with the capillary beds in the lower epidermis and deeper dermis. In fact, some salamanders lack lungs and depend entirely on cutaneous respiration through the skin to meet their metabolic needs. Cutaneous respiration. The most primitive tetrapods had scales like the fishes from which they arose. Among living amphibians, dermal scales are present only as vestiges in some species of tropical Sicilians, Apoda. Frogs and salamanders lack all traces of dermal scales. In salamanders, the skin of the aquatic larva includes a dermis of fibrous connective tissue consisting of a superficial, loose tissue over a compact, deep layer. Within the epidermis are deep basal cells and surface apical cells. Scattered throughout are large Leydig cells thought to secrete substances that resist entry of bacteria or viruses. In terrestrial adults, the dermis is similarly composed of fibrous connective tissue. In the epidermis, Leydig cells are now absent, but distinct regions can be recognized such as the strata basal, spinosum, granulosum, and corneum. The presence of a thin stratum corneum affords some protection from mechanical imbrasion and retards loss of moisture from the body without unduly shutting off cutaneous gas exchange. During the breeding season, nuptial pads may form on digits or limbs of male salamanders or frogs. Nuptial pads are raised colossus of cornified epidermis that help the male hold the female during mating. The skin of frogs and salamanders includes two types of multicellular glands. First is the mucus and second is the poison glands that are both located in the dermis and open to the surface through connecting ducts. The mucus glands tend to be smaller, each being made up of a little cluster of cells that release their products into a common duct. The poison glands tend to be larger and often contain stored secretions within the lumen of each gland. Secretions of poison glands tend to be distasteful or even toxic to predators. Few persons handling amphibians are bothered by these secretions because it is potentially harmful only if eaten or injected into the bloodstream. The skin of reptiles reflects their greater commitment to a terrestrial existence. Keratinization is much more extensive and skin glands are fewer than in amphibians. Scales are present, but these are not fundamentally different from the dermal scales of fishes, which are built around the bone of dermal origin. The reptilian scale usually lacks the bony undersupport or any significant structural contribution from the dermis. Instead, it is a fold in the surface epidermis, an epidermal scale. The junction between adjacent epidermal scale is flexible hinge. If the epidermal scale is large and plate-like, it is sometimes termed as scute. Additionally, epidermal scales may be modified into crests, spines, or horn-like processes. The dermal bone is present in many reptiles although they are not usually associated with scales, like for example the gastralia, a collection of bones in the abdominal area. Where dermal bones support the epidermis, they are called osteoderms. They are the plates of dermal bone located under the epidermal scales. Osteoderms are found in crocodiles, lizards, and some extinct reptiles. Some bones of the turtle shells are probably modified osteoderms. The dermis of reptilian skin is composed of fibrous connective tissue. The epidermis is generally delineated into three regions: the stratum basal, stratum granulosusum, and stratum corneum. In turtles and crocodiles, sloughing of skin is modest, comparable to birds and mammals, in whom small flakes fall off at irregular intervals. But in lizards, and especially in snakes, shedding of the cornified layer, termed molting, results in removal of extensive sections of superficial epidermis. As molting begins, the stratum basal, which has given rise to the strata granulosusum and corneum, duplicates the deeper layer of the granulosusum and corneum, pushing up the old layers. White blood cells invade the stratum intermedium, a temporary layer between old and new skin. These white blood cells are thought to promote the separation and loss of the old superficial layer of the skin. Integumental glands of reptiles are usually restricted to certain areas of the body. Many lizards possess rows of femoral glands along the underside of the hind limb in the thigh region. Crocodiles and some turtles have scent glands. Sample, in the alligator of both sexes, one pair of scent glands opens into the cloaca, and another pair opens into the margin of the lower jaw. In some turtles, scent glands can produce quite pungent odors, especially when the animal is alarmed by handling. Most integumental glands of reptiles are thought to play a role in reproductive behavior or to discourage predators. Birds' basic structure. The feathers of birds have been called nothing more than elaborate reptilian scales. Certainly, the presence of epidermal scales along the legs and feet of birds testifies to their depth to reptiles. If not a directly modeled reptilian scale, then the feather is an example of yet another more fundamental homology of the underlying interaction of the epidermal dermal layers producing such a skin specialization. During the brooding season, the dermis in the breast of some birds have become increasingly vascularized, forming a broad patch in which warm blood can come into close association with incubated eggs. The bird skin has few glands. First is the epidermal gland, which is located at the base of the tail that secretes a lipid and protein products that bird collects on the sides of their beak and then smear on their feathers. The other gland located on the heads of some birds, the salt gland, which is well developed in marine birds that excrete excess salt obtained when these birds ingest marine foods and seawater. Salt excretion. Feathers distinguish birds from all other living vertebrates. Feathers can be structurally elaborate and come in variety of forms. Yet feathers are non-vascular and non-nervous products of the skin, principally of the epidermis and the keratinizing system. They are laid out along distinct tracts called terrellae on the surface of the body. They are replaced each year via one or several molts. The modern bird feather is built from a tubular central shaft, which is the rachis, which carries on either side a vein and a series of barbs with interlocking connections termed barbules or the hooklets. The rachis and attached veins constitute the spathe. The rachis continues proximal as the barbels, calamus or keel, which anchors the feathers to the body and often removed by attached dermal muscles. In modern birds, feathers are of many types serving different functions. Flight feathers are long and the veins asymmetrical about the stiffening rachis. Those flight feathers on the wings are remiges or remix, and those on the tail rectrices or rectrix. The contour feathers or pinnaceous feathers covers the body and usually have symmetrical veins about rachis. Down feathers or plumulaceous feathers lock a distinctive brachis and non-interlocking barbs extend out from the calamus as a fluffy feather that is important in insulation. Feathers develop embryologically from feather follicles, invaginations of the epidermis that dip into the underlying dermis. The root of the feather follicle, in association with the dermal pulp cavity, begins to form the feather. During feather regeneration, inductive interaction between the dermal papilla and base of the follicle establishes a zone of cell proliferation, where new keratinocytes are produced, and a zone of patterning just above it that will generate the morphogenetic signals presiding over the fates of these keratinocytes. In the follicle, rings of outer cells, the beta-keratin, the sheath and feather itself form more or less concentrically around the inner stratum cylindricum and pulp cups, alpha keratin, and the dermal core. The feather filament continues to grow out from the follicle accompanied by the highly vascularized dermal core, which extend through the follicle mouth above the surrounding integument. Functions of several types of feathers. Contour feather aerodynamically shape the surface of the bird. The down feathers lie close to the skin as thermal insulation. Developed plumes are often specialized for display, and flight feathers constitute the major aerodynamic surfaces. Flight feathers of the wings are of type of contour feathers. They are characterized by a long rachis and prominent veins. These feathers have some value of insulation, but their primary function is locomotion. Most feathers receive sensory stimuli and carry colors for display or courtship. Chromatophores occur within the epidermis, and their pigments are carried into the feathers to give them color. Evolution of feathers. When we think of feathers, we think of their roles in flight, but they likely had other functions when they first arose. One view is that feathers or their scaly predecessors played a role in surface insulation. Surface insulation, of course, holds heat in or shields the body from taking up excess heat. Surface insulation would have interfered with the absorption of environmental heat, a disadvantage if the ancestors of birds were ectothermic. However, many species of ectothermic lizards have enlarged surface scales. Once the best king lizard is warmed, it turns so that the scales act like many tiny parasols to shade the skin surface and block further uptake of solar radiation. Once enlarged and shaped for heat exclusion, these protofeathers would be pre-adapted for heat retention or for flight. Others argue that ancestors of birds were endothermic. In this view, protofeathers initially functioned to conserve internally produced body heat. The evolution of aerodynamic devices serving flight came later. Whether beginning with an ectothermic or an endothermic, many still suggest that feathers played a role in surface insulation when they first appeared and became secondarily co-opted for a role in flight. However, early birds and their immediate dinosaurian ancestors lacked nasal turbinates, a character diagnostic of warm-blooded physiology. If serving as insulation, these first feathers would have a more complicated role than previously thought. Dinosaurs had to call the sequel. A different view entirely stems from argument that feathers evolved initially as aids to gliding and then to flight. Feathers were selected because of their favorable effect on the airstream passing over the body or limbs of a gliding animal. If the protoavian limb were not streamlined, then pressure drag would result, and turbulence would have reduced aerodynamic efficiency. However, surface scales projecting from the trailing edge of the limb would have streamlined the limb, reduced drag, and thus been favored by selection. Aerodynamic principles. Regardless of whether they evolved first for gliding or for insulation, feathers were modified from reptilian scales, or at least from the common inductive interaction between dermis and epidermis. In modern birds, feathers that serve flight are highly modified. Interlocking barbs and barbules give some structural integrity to the flexible flight feather. In flight feathers, the rachis is offset, making the vane asymmetrical. This design affects the action of the flight feathers during the wings beat. In the downstroke, the pressure of the underside of each feather acts along its anatomical midline, the center of pressure. But because of the rachis is offset, the result is to twist the feather slightly about its point of attachment to the limb, forcing feathers of the wing together into a broad surface, then that presses against the air and drives the birds forward. On the upper stroke, the center of pressure is now across the top side of the asymmetrical feather and forces it to twist in the opposite direction, opening a channel between the feathers. This reduces their resistance to the airstream and allows the wing to recover and prepare for their next power downstroke. Ruffled feathers. The figure shown is the feathered dinosaur. In Figure A possible reconstruction of an early feathered theropod was shown. In B, this is the summary of phylogeny showing current claims for the occurrence of feathers in theropods. In some taxa, only a few species shown evidence of feathers. Earliest to appear are the down feathers, then contour feathers, and asymmetrical feathers or the flight feathers, then followed on by the feathers of the forearms, W. Bird flight. This controlled twisting of flight feathers, possibly responding to wing beat, depends on the asymmetrical design of the feathers and hence on the action of air pressure against it during powered flight. Here is a figure of Archaeopteryx feather that also reveals offset rachis and an asymmetrical vane. This feather from the wings of Archaeopteryx shows the asymmetrical design of the vane, suggesting that it may have been used during powered flight as in modern birds. The epidermis of the mammals may be locally specialized as hair, nails, or glands. The epithelial cells of the epidermis are keratinocytes and belong to the keratinizing system that forms the dead superficial cornified layer. The surface keratinized cells are continually exfoliated and replaced by the cells arising primarily from the deepest layer of the epidermis, which is the stratum basal. Cells within the basal divide mitotically, producing some that remains to maintain the population of stem cells, and others that are pushed outward. As they are displaced to a higher level, they pass through keratinization stages, which is the stratum spinosum, stratum velanosum, and often stratum lucidum, and the stratum corneum. Keratinocytes, or the process of keratinization, is most distinct in the region of the body where the skin is thickest, as on the soles of feet. Keratinocytes are the most prominent cell types of the epidermis. Other types may be recognized, but they are less clearly known. The Langerhans cells are stellate cells dispersed singly throughout the upper parts of the stratum spinosum. Current evidence suggests that they play a role in cell-mediated actions of the immune system. The Merkel cells, originating from the neural crest and associated with nearby sensory nerves, are thought to respond to the tactile stimulation, or the mechacoreceptors. In addition to these epithelial cell types, the other prominent cell type that becomes secondarily associated with the epidermis is the chromatophore. Chromatophores arise from the embryonic neural crest cells and may be found almost everywhere within the body. The mammalian dermis is double layer. The outer is the papillary layer that pushes finger-like projections, termed dermal papillae, into the overlying epidermis. And the deeper layer, reticular layer, includes regularly arranged fibrous connective tissue and anchors the dermis to the underlying fascia. Blood vessels, nerves, and smooth muscles occupy the dermis but do not reach the epidermis. The mammalian epidermis produces dermal bones, but these contribute to the skull and pectoral girdle and only rarely from dermal scales in the skin. One exception is the glyptodon, a fossil mammal whose epidermis was underlaid by dermal bone. A similar situation exists in the living armadillo. This species represents secondarily developments of dermal bone in the mammalian integument. Blood vessels and nerves enters the dermis. Hair follicles and glands projects inward from the dermis. Hairs are slender, keratinous filaments. The base of the hair is the root, and its remaining length constitutes to the non-living shaft. The outer surface of the shaft often forms a scaly cuticle, and beneath this is the hair cortex, and at the core is the hair medulla. The hair shaft projects above the surface of the skin, but it is produced within an epidermal hair follicle that is rooted in the dermis. The surface of the epidermis dips down into the dermis to form the hair follicle. At its expanded base, the follicle reserves a small tuft of the dermis, which is the hair papilla. This papilla seems to be involved in stimulating activity of the matrix of the epidermis, but does not itself directly contribute to the hair shaft. The tiny clump of living matrix cells, like the rest of the stratum basal, is the germinal region that starts the process of keratinization to produce hair within the follicle. Unlike keratinization within the epidermis, which is general and continuous, keratinization within the hair follicle is localized and indeterminate. The shaft of hair grows out of the living hair follicles, which go through a cycle of activity with three stages: the growth, degeneration, and the rest. During growth, there is an active proliferation of cells in the hair papilla and the base of the hair, producing successive addition to the hair shaft, which emerge from and continues to lengthen from the skin surface. At the end of the growth stage, the hair-producing cells become inactive and die, so it enters the degeneration stage. Thereafter, the follicle enters the resting stage, which may last for several weeks or months. Eventually, stem cells in the papilla produces a new follicle and the growth stage begins to gain again. A thick of covering hair, fur, or pelage is generally composed of guard hairs and underfur. The guard hairs are large, coarse hairs that are most apparent on the outer surface of the fur, and the underfur is stationed beneath the guard hairs and is usually finer and shorter, but they both function as largely as insulators. In most marine mammals, the underfur is reduced or lost entirely, and only few guard hairs are evident. Some hairs are specialized. Sensitive nerves are associated with the roots of the vibrissae, or whiskers, around the snouts of the mammals. Such a sensory function may have been the first function of hair in the form of vibrissae, appearing before in an insulative pelage evolved. Vibrissae are common today in nocturnal mammals and in mammals that live in burrows with limited light. The quills of porcupines are stiff, coarse hairs specialized for defense. Evolution of hair. Fossil skin impression from the Middle Jurassic give evidence for the presence then of hair and presumably of mammals that owned it, but the initial adaptive value of hair remains speculative. One view holds that hair arose initially as surface insulation, which retains body heat in primitive mammalian endoderms. The presence of nasal turbinates in the synapsids earlier from the late Permian suggests an early endodermy and hence a role for insulating hair. An alternative view is that the hair evolved first as tiny projecting rods in the hinges, between scales and serve as a tactile devices. Being soft and decomposable, hair does not leave a reliable trace in the fossil record. Some therapsids, ancestors to mammals, have tiny pits in the facial region of their skulls. These pits resemble pits on skulls associated with the sensory vibrissae in modern mammals. Some have interpreted that these pits is an indirect evidence of hair in therapsids, but the skulls of some modern lizards with scales have similar pits, but of course, lizards don't have hair. Such pits are not conclusive evidence of presence of hair. There are three main types of integumental glands in the mammals, namely the sebaceous, exocrine, and apocrine. The sebaceous glands produce an oily secretion, sebum, that is released into hair follicles in order to condition and help waterproof fur. Sebaceous glands are absent from the palms of hands and soles of feet, but they are present, without associated hair, at the angle of the mouth, on the penis, near the vagina, and next to the mammary nipples. At these sites, their secretion lubricates the skin surface. The wax glands of the outer ear canal, which secrete earwax, and the meibomian glands of the eyelid, which secrete an oily film over the surface of the eyeball, probably are derived from sebaceous glands. The screen glands produce thin, watery fluids that are not associated with hair follicles, begin to function before puberty, and are innervated mainly by cholinergic nerves. In most mammals, these glands are associated with the soles of the feet and hands, prehensile tails, and other sites in contact with abrasive surfaces. The apocrine glands produce a viscous, lipid-containing fluid that are associated with hair follicles that begins to function at puberty and are innervated mainly by adrenergic nerves. Their secretion primarily function in chemical signaling. Cholinergic and adrenergic innervation. The scent glands are derived from the apocrine glands and produce secretions that play a part in social communication. These glands may be located almost anywhere on the body: on the chin of some deers and rabbits, on the face of some deers, antelope, and bats, in temporal regions of elephants, in chest and arms of many carnivores, anal regions in rodents, dogs, cats, mustelids, belly in most deer, back in the kangaroo rats, peccaries, camels, ground squirrels, or legs and feet in many ungulates. Secretions of these glands are used to make territory, identify the individual, and communicate during courtship. The mammary glands produce milk, a watery mixture of fats, carbohydrates, and proteins that nourishes the young. Ectodermal mammary ridges, within which mammary glands form, are located along the ventrolateral side of the embryo. The number of mammary glands varies among species. Release of milk to suckling is lactation. Lactation has been reported in males of a species of Malaysian fruit bat, opening the possibility that these fruit bat males may actually suckle the young they sire. Outside of this fruit bat, lactation has been reported only in domesticated male animals. The likely resulted of abnormal inbreeding and in pathological conditions. With these exceptions, mammary glands became functional only in females. Mammary glands consist of numerous lobules, and each lobule is a cluster of secretory alveoli in which milk is produced. The alveoli can open into a common duct that in turn can open directly to the surface through a raised epidermal papilla or the nipple. This nipple is usually surrounded by a circular pigmented area of skin called the areola. Alveolar ducts also open in a common chamber or cistern within a long collar of epidermis called the teat. The teat forms a secondary duct carrying a milk from the cistern to the surface. Adipose tissue can build up beneath the mammary glands to produce breast. Daniel Blackburn led to hypothesize a series of steps in the evolution of mammalian lactation, beginning in ancestors who incubated eggs or hatchlings. The skin gland secretions of the females that had antimicrobial properties would protect the surface of the young from bacteria, fungi, or other pathogens. Ingestions of these secretions in small quantities by hatchling would reduce pathological effects and microorganisms in the digestive tract. If this secretion include maternal immunoglobulins, this would also directly deliver immunity to the offspring, or any nutritional value included with these secretions of ancestral mammary glands would be of further adaptive significance. In maternal skin glands of incubation patch first supply protection from pathogens and then secondly become involved in nutrition of the young. Subsequent evolution includes enhancement of the secretion into a copious flow that was much more nutritious.